introduce Roseanne Miner. She was the diagnostician at Komohana Research Center for quite a while, and also working as
1 year, 10 hours.
well, it felt like a long time.
Yeah.
You did so much for me, but happily retired?
Yes.
Okay, happily retired. She was also working on the coffee berry board team, and that's how I know Roseanne, but she's got an extensive history. So I think I'll let you talk a little bit about it, Roseanne.
And I won't tell you the whole thing. So I'll start. I'll take questions during. I'd rather, have you ask the question before you forget, because I've learned at this stage in life that sometimes I can't remember as fast as I'd like. And so my question, and a long time ago for a group of students at high school, is how many years do you think I went to college? And I'll go up or down. Take guesses. 15. Down a little. 6. Up. 13. Up. 14.
Wow.
And then the rhetorical question, that's don't answer this. Does that make me smart or dumb? 14 years, but I have worked at universities pretty much for my whole adult life. This is at the very start of working, but I had come there with a degree in plant science to do integrated pest management. So this is, if you wanted to summarize my life in one slide, we have Alaska, and my husband, he 25 years on a small vegetable farm. So from living there as well as working in it.
Is that another municipality?
Yes. We were in between Palmer and the state fairgrounds was in between my farm and Palmer. But the other part to this, which I can't see on here, so I'm going to walk over and point out, at a certain point when I was working as a tech, I started working with plant diseases because, insects, you have weeds for competition, but plant diseases are like the last and the hardest. But from a person who likes ecology, you have the interaction of two organisms. So it's like, how does one get the upper hand? If something rots, the fungus harvested it before we did, is what I say. So this is a lettuce plant in the top right, and there's white mold on the right there. It's a fungus that I could tell you the name of, but I won't do that right now. And you can see water soaked and then healthy tissue. So that was that part of, if I'm going to be a diagnostician, I would isolate right from the middle of that slide, take a little piece and see what grows. And I grew thousands of petri dishes of that and stuff. So the growers like, why do we have this? Why does it spring up in the middle of the season? And they actually, the life cycle hadn't been there before. So this is the next picture. The little disc over there is actually called the discomycete. It releases spores out of that little plate, and those spores go over and land on the edge of the lettuce. So if the lettuce has a little tip burn or some other thing, it's a problem. Flower petals are very susceptible to the spores. So one of the things I told them is when you see these in the field, then, decide what management you're going to do. There was a case of canola oil, and they said that they studied this one pesticide, and they also studied weed control. And they said that weed control, keeping the weeds out with more air, was as effective as the chemical control in that case. So plus, if you do weed control, you don't have the weeds in the seed bank. This happens to be pineapple weed, which is really nasty in Alaska. It just forms a mat, and you pull up all the soil with the roots. And then on the bottom here, a wild rose. On the bottom, there was a rose hip starting, and then a young one. So I was telling the photography club last night that that's my best picture of a wild rose in 30 years of trying. Okay. But now you've heard a different version of me because I haven't had pictures in the past years. And this talk, very similar to last year. I reversed the order from the first year I gave it, and Brian is the one who set up this slideshow. So we'll talk about him. He had 20 plus years of working as a diagnostician. I can tell you about cold weather crops, but you guys probably know about gardening even more than I do in the tropics. I said I'll spend the rest of my life trying to learn all the tropical plants.
So you just have to click left.
Like this one?
Yeah.
Okay. And can I, oh yeah, this is very helpful.
Good.
Yes.
Maybe I'll sit right here.
So Russell said he wanted me back after 2 years of crying. And I would say that my style of presentation, I taught college biology for a while. I taught 2 times plant pathology in college, but I also worked at a nature center before I went to college or during college. And that part you have to be more enthusiastic and less formal. So my style is different. And, you know, if it helps today, disease management and island ecology is in front because this is similar for so many of the plant diseases. I'm having fun here. I guess I have to go a little slower. Basic phytopathology, that's in the middle of the ground rules. And keep in mind that, you know, 7.5 years for the PhD. So, you know, I could talk a lot about, you know, but the basics. And plant disease symptoms, these are last, and there's far more fungal diseases than these others, because what I'm going to do is give you a quick introduction. You will have the internet, you will have books, you will have other resources. So here you'll get familiar, but as somebody brings in and has a question, you'll look it up with more current information. I don't want you to memorize any names of these causal agents because you'll find that out later. You know, there used to be a time in education where it's memorized this, and now it's just nowhere to look it up. Okay. So when I worked with the coffee berry borer, there was a person who grows coffee and studies coffee berry borer in Ka'u, and he said, I put the conclusions first. So if you don't get to the end of the talk, it's okay, because the best part was first. It'll be mentioned again at the end, but to remember that a garden change is constant and it's a cycle. So that part of, you know, it's going to grow. If it's not a vegetative part like a lettuce plant or baby greens or other things, it's going to bloom like a tomato or coffee, and then the fruit is often harvested. But this part of, you know, look again next week, look in 10 days, the idea that things stay the same is always contradicted by a garden. So remember that, you know, it's not you go look once, you're looking a lot. And I'm gonna have to keep the mouse here. Decay and harvest. In my case, I literally say that the fungus harvested it before we did. You know, in the terms of ecology, it's like, where did the energy go to? We didn't get the energy, another organism did. So that would work well out in the woods. Thank goodness there's things that decay the leaves that fall off and the branches and the other stuff, or you wouldn't get rich soil. But when they do it in a garden ahead of us, it's not as much fun. So, diagnosis summary. You know, I'm trained in science and science questions. It's not that I have all the answers or other scientists have all the answers. It's actually, can I ask questions with the best of them? And I have told Russell, my granddaughter, I see her on Zoom more than in person, and she wanted to play a guessing game. And I realized that this is the best science training I can give her. Is like, well, what is it? Is it that part about when you have a garden problem, you start asking questions, knowing that you can't answer all of those questions. But a scientist, you know, where's Waldo? What's wrong with this? Why isn't this expected? What would be the how will this change if I add something? All of these questions are good. Some of them can be answered by science. Why do I like it? Does it smell good? Those are questions that probably can't be answered by science. So there's still valid questions, but usually in diagnosis, I'm going to look for science-based, either qualitative or quantitative questions, you know, that maybe I can answer to help with letting us harvest the plant instead of something else. But all of these questions, do you think it's pretty? That's a lovely question. And when I taught master gardeners in a little bit in Alaska, I was saying like house plants, and they're like, oh, I can't grow orchids, they're too hard. I'm like, well, can you grow an African violet? And if they said yes, I'm like, then you can grow an orchid. And if they said no, I'm like, well, maybe you want to work on African violets first. But how do you grow African violets? Well, you put them where they look that they're happy. They don't look happy, you move them. And they're like, define that. How do they look? You know, are there leaves? So that part of it's not scientific to say it that way, but I think you understand what I mean. And when you're talking to people, you know, they might want the science end of it. They might want the look happy end of it. All good. Do what works. Educated guess. Now, sometimes people say I have a theory, you know, in the Big Bang theory. In science, theory means something that is supported by data from many experiments. So I just, hypothesis is an educated guess for one experiment. And that makes better sense to me. But anytime you have these questions, the part of, okay, if I try this, what do I think will happen? And if you're doing good science, usually what happens is not what you expect. But at least you have data to show that. So you guys will be looking up data that other people took that may have surprised them. Education, finding relevant information. I would just advise you to look for most websites that say .edu. You know, there's lots of blogs and some of them are wonderful, but the edu ones usually have more scientific background to go with them. So that part of, if you're doing it as a master gardener, then you want to find information that is reliable. So change is constant, we did. And this is different from Alaska, where we had all this space and a few plant species. I mean, literally, tree genuses on one hand. So that part of, and things came up, they were killed by the frost in the wintertime. So here is a whole different, you know, we have, one of the saddest things to me is rapid ohia death, because this is 80% of the native forests, and I live in Puna just for 5 years, and I've literally watched ohia trees die in my yard. Yeah, so that part of how can you be good ambassadors for helping yourself and other people be more protective of this ecosystem? It's all of us a responsibility. So for an island or for a disease, you know, your greenhouse, your yard, that you could call that a little island. You know, the first thing you want to do is exclude anything you can. So when I grew orchids in the house under lights, you know, if I got a new orchid, it was 3 months before it could go sit with the others, because I needed to know that there were no bugs, there was no other things. I didn't want to carry it back to plants that I'd had for 10 years. So being really choosy with what you bring home. You know, if it doesn't look good, if you find something when you brought it home, can you identify it? You know, the websites now are really good. You know, you have the master gardeners to help you. If if you're a commercial grower and it's going to cost money or it's not listed, the diagnostician here can help with that. So, some are easy, some are hard, but you probably want to know what you're working with. And then managing, different than control, because a lot of management is what used to be called common sense. How's the air circulation? You can get rid of weeds and have better air circulation. That part of how much water, how much sunlight. We just had a discussion. I was saying that I had a phalaenopsis from Christmas sitting in the middle of my house. And eventually it got crown rot and I had to throw it out. I should have put it outside where it got more light sooner. So, I'm talking about diseases, but the lack of sunlight, uneven watering, these are even bigger. And if you talk to farmers in vegetable fields like I did, it's not the diseases come in at the end, it's the weeds that come in and compete with the seedlings. So the disease is like the icing on the cake. Certainly want to prevent it if you've gotten it that far, but light, water, competition earlier. So control. A lot of people think that, as a disease specialist that I work with fungicides. I have, but usually that's the last line of defense, and you've almost lost the battle if that's where you're at. But on the other hand, there are times where it's very judicious. So if you're going to go to chemical control, you want to know exactly what you're working with and have the right amount and make sure you're not affecting, plants outside the treatment area, especially, neighbors and stuff like that. So I picked these pictures. They were taken by Brian. They were in his slideshow. And the question is, what do you think it's causing this? So now I'll give you a better hint. You'll probably only see it in South Puna, especially in 2018.
Sulfuric acid.
Yeah, so this fog damage, and it's pretty hard to exclude, basically you wait it out and try again. If you can move small specimens, but this being a case where there wasn't a lot to help. So the integrated approach, this again, thanks to Brian, physical methods. Never underestimate sanitation barriers exclusion are the best first start. On this island, please just don't even bring it here. That's our best thing. Coffee berry borer wasn't here till 2010. And, the little borers get into the berries and ruin them for coffee. But while I was working on the grants, 2020 to 2022, we had coffee leaf rust come to the state. It took coffee berry borer 10 years to reach all the islands. It took coffee leaf rust 6 months. And coffee berry borer ruins the berries, but it does not harm the tree. Coffee leaf rust, if you get 20% of the leaf covered, the plants drop them. And if you drop too many leaves, you don't have enough energy. So it's far more significant that this one plant disease is finally here. But it's also in other places where they grow coffee. And if you get all the details right, you can have a profitable crop. It's just harder to manage more details and probably a smaller profit margin because of the extra inputs for control. So if possible, sanitation first. In the case of Alaska, potato growers I work with a lot, and there was a disease called late blight, which actually caused the Irish potato famine on the wind. That one of the biggest things, well, in Alaska, like don't let it get here. And it usually dies out in the winter. But if it gets there, where's your cull pile? It's right next to the barn, blowing onto all the stuff. So if you have a compost heap, there's certain things that if I have them in the house, they go to the transfer station, they don't go to my compost pile. If there's any chance that your compost pile isn't being turned regularly enough, that's a consideration. The other part here, raised benches. Turmeric used to be grown in the ground, and now there's a bacterial wilt that raised benches is better because that's a way to exclude it from the root zone. Once it's around in the ground, footprints, anything that tracks it from one place to the other, it's easier to grow it if you could grow it in the ground, but it's more reliable to get a yield if you grow it in a pot and raised benches. Clean culture. Okay, what's next? Biological methods. There's lots of beneficial organisms. For taking the coffee berry borer as one, it's being sprayed with a fungus that hurts the insect. So, and that fungus is naturally occurring, but it's commercially formulated to get it in the right place at the right time in excess, so that what's out there in the wild is not going to be enough of a balance. But you can shift the balance. Compost tea, biopesticides that I was just talking about for coffee berry borer, and then pathogen-free plants. Again, be picky about what you bring. I think that some of the hardship in Puna is it's where the landscape industry is because we get so much rainfall, but sometimes you bring in plants in pots and you can't tell what all is in the pot along with the plant. Coqui frogs, fire ants. It's just, yes.
Back to your physical methods to raise benches and
Yes.
saying bags. So as the leaf wilt, the bacteria as the leaf wilt flows out of the raised bench down onto the ground, is that not a problem?
Not if you're not doing stuff on the ground.
Okay.
So that it's,
Sorry, we didn't hear the full question. Would you mind repeating it?
okay, yes, that's a good plan. So he was asking with raised benches under physical methods, if you water the pots, whatever's in the pot goes to the ground, and wouldn't that be a problem? Hopefully it's not in the pots. And if you see some wilt in the pots, you remove it far away. And whatever goes to the ground is not supposed to be back up in touch with the raised benches. So it's really just a physical, yes, a physical exclusion. The part of, there's a lot of other things in the soil that compete, so that one bacteria that gets in there doesn't have a free ride down there. But if you touch your shoes and touch the plant pot, you possibly could spread it back up. And when the coffee leaf rust is going to be my favorite example, it was in Kona before it was in Ka'u. So, spray off your truck, wear a Tyvek shoe, do this to your shoes. We do this for rod as well. If you go hiking, you go with clean shoes, so you're not bringing it to a place where it's not there already. Probably, though, a bigger thing is pigs are bringing rod and they're wounding the roots.
Yeah.
So the pathogen is not that strong, but if it's right there as the wound is made. So we digressed a bunch. I love talk story. And if somebody from a different place would like to ask questions on Zoom, please let me know.
I have a question.
Okay.
Just I'm curious, when you're talking about the difference between the coffee borer beetle and how long it took, 10 years, and the leaf rust 6 months, and it's everywhere. So what just different pathogen, or what was making this tremendous difference in spread?
That's an excellent question. And think of this, the coffee berry borers are pretty little, but they're still big enough that they won't blow on the wind. It's as far as they can fly or get carried in coffee beans. So it was actually island to island spread was probably in somebody's luggage or product. But coffee leaf rust has teeny little spores that look like rust on the underside of the leaves, and it blows on the wind. So JB Friday, who works with trees, he went to a hike, YPO Valley, up from that, Waimanu, and there was a lot of volunteer coffee there from past times, and it had coffee leaf rust. You know, it had blown by itself. So biological, it's fun, but biological is probably also in shades of gray. It's never going to be a complete cure for what's there. So you're just keeping the balance of power in the plant's favor so you can harvest it. Host resistance, that's actually, plant breeders have done so much with that, and they usually do it with traditional breeding. You know, now some people have used genetically modified, but that part about traditional breeding is usually the source of a lot of host resistance. Yep, chemical methods. Fungicides, bactericides. Side means something that kills. So fungicide to kill fungi, bactericides, you know, agaricide is killing mites, herbicide is killing weeds. And there was, after World War II, a lot of people took this as a wondrous thing without understanding the ramifications. So in 1960, Rachel Carson, Silent Spring, you know, the birds are being affected by what you're doing with a pesticide. So I think that people are much more careful about how they use them than they used to be. And I'm very glad of that. So this was Brian's first slide, and it just had his name. The Agricultural Diagnostics Service Center, ADSC, is on Oahu, and Darren Park is in charge of it. But Brian worked over here because why send disease samples to Oahu when it might not be there yet? It's very helpful to have Brian here. Now, when Brian was ready to retire, I was sitting down at that picnic table, and Brian's like, nobody's going to replace me. And I'm like, oh, I used to do that. So and that was November. And, you know, by December, a month later, I was hired for 10 hours a week as a stopgap. And, you know, I told Darren, you know, I'm on a temporary hire. It was extended for a year, but they didn't extend it more than that. And also it was more than I wanted to do. You know, I had 20 hours a week with the grant and then 10 more hours. And so Gabriela Benito was hired last April. She's been doing it for a year. She worked with Rod before this with the Forest Service. So she has a lot of knowledge that she brings, but as somebody was saying, you know, you have to work to grow into the shoes that Brian left. You know, he had 20 years, he could look at something, you know, he knew how to culture it. He allowed me to send him emails with pictures saying, Brian, what do you think about this? And you will find out as master gardeners, you know, ask the people, you know, somebody might give a hint that helps everybody. So please, you know, have conversations with other people that have different experiences than yours. But you know, there's been 3 people here. There was also on Manoa, it is more soil testing as well. And they had the seed lab. I'm not sure if they still have money for that and other things. So that part of the size of the industry and the size of the university and the effect of the pandemic as to how long what services will be available. But Gabriela is doing a lovely job. And she's here and I'm not. This is the form that we have to fill out. And the reason I show it to you guys, that on a few occasions, we've received a box that somebody was sending us a sample. Is that okay? Is it okay to mail disease samples without a permit? You know, you could be mailing coffee berry borer or, you know, the first time that I just started, Brian had a sample and we were looking through the plastic bag. It's like, that's coffee leaf rust. And Lisa Keith over at ARS already looked at it. It's like, we're not even going to open it. We're just going to autoclave it because we didn't want to accidentally spread it on Hilo's side. You know, that part of thinking in those terms, I was saying to be good at culturing fungus, basically good sterile technique, tissue culture is paranoia. If you're too careful, no problem. If you weren't careful enough, you can't put Pandora back in the box, you know, the cat out of the bag. This permit is valid for 1 year. So Darren has to work to ask and get this every year. So if you're bringing in a sample, if it comes here and Gabby's on station, she's out of state right now, then it doesn't have to get mailed anywhere. But if it's a soil sample, you still can have nematodes and other things along with that soil. So it can't get mailed except by an extension office. So please, you guys probably knew this before I told you, but you might be able to tell some of the people that come here that, oh, can I just mail it myself? I'm like, and they will accept it in Waimea, they will accept it in Kona, so they don't have to drive to Hilo to drop it off. But that part about, please help us keep this permit, help other people know that they shouldn't, if there's actually a protocol, if I get a box that shouldn't be there, you know, pretty much it's going to get autoclaved, and autoclaved is a big pressure cooker.
Yep.
So on the top left is a sample for a disease sample. That's really too late. I can't tell what's left. Maybe if it was a soil sample from a greenhouse, but again, I want to credit Brian. You know, don't be part of the problem. All of these things have come. Ohia rust, you know, I've personally watched a little Ohia die that my son won at a contest. And it was just, it's like, I see it, but it's too late now. And Ohia rust, you know, I'm not sure they know how it came here, but it hurts the little Ohias. And some are more susceptible than others. So some will, but what I noticed is I put that little Ohia plant next to a rock wall where there was less air circulation. So the next ones, they're getting all the wind I can give them. And was it because they were more resistant or because I changed the environment? So all of these things, banana bunchy top, you know, I can read about when they were trying to eradicate it and they were asking people in Kona to, if we find it at your place of residence, we want you to kill this banana plant. And some people certainly didn't want to do that. But if you want an industry to grow bananas, you can't have the aphids carrying it from your backyard elsewhere. So Brian did tell me that he was very happy to work with master gardeners because they made his job easier. So all of you, please, you're the front line for the university to say, you know, think about that. Is there a common sense reason that might not be a good idea? You know, you are very helpful in all of these. And while we have more bugs here, actually most of the questions that come for diagnosis are plant diseases. And even what I look at today, I can't do what I did in the lab because I had a microscope and I had culture plates. So that part of, I don't even expect you to diagnose diseases the way I do because I can't do it without specialized equipment. So that's where you can say the symptoms or the signs look similar. You know, if you wanted to, you could pay to get it tested, or you probably just want to do sanitation and remove it. And so time and money and everything else. So the best, I'm going to emphasize this as much as possible, that science questions are better even than answers. If you don't even ask the question, then you might be missing the point. Experience is cumulative. So, Brian learned all of this stuff over time. I don't have to work as hard. I just read what Brian wrote, what other people wrote. I guess the other part to that is that it also changes with time, that changes constant bit, that they change the names of a lot of fungi. They even changed the names of plant families. The plant family for daisies used to be Compositae when I was going to school in, my lifetime in the 80s. And now it's Asteraceae. So it matches better. And like, fine, now I just have to know two, because in the old reference, it'll say composites, and the new, it'll say Asteraceae. But that part of, and here, if I'm learning plants, if I talk to JB, he wants me to say the scientific name. If I'm talking to my neighbors and stuff, they want me to say the local name. And like, if we take Pandanus, screw pine just is not a common name that I want to use much, but Hala, the Hala trees that are out in front of. So now there's the common name, the scientific name, and the Hawaiian name. To be very respectful, try to learn as many of them as you can. Classify large and small. I was saying how plant families change, organisms change, but when a little before I went to school, there was just two kingdoms, plant and animal. And then they said that, well, bacteria, are they little plants or little animals? It's like, okay, they're another kingdom. So this is big classifications. And then they realized that some things like algae, well, it's not a bacteria, but it's not. So then there was the protozoa. And about the time that I was in college in the 80s, a little sooner, they made fungi the fifth kingdom, because fungi were not little plants. They were not as simple as protozoa mostly. And, they weren't single-celled like bacteria. And then when I taught biology in the late 90s, they said, okay, there's kingdoms, but that we found out with DNA studies that bacteria are not one big group. They're actually the Archaebacteria and the Eubacteria. They may have changed it since then, but the Archaebacteria live in hostile places and are more likely to have generated multicellular things, maybe. And then the Eubacteria are very common, but they probably don't have as long a history. And so this whole part of not only in the garden is change constant, but in the scientific literature and stuff, it's like, roll with it, please. Source credibility, again, .edu, but don't discount when somebody says, oh, well, there's this trend. It's like there was a comment, wives' tales, at a lot of those, there's a kernel of truth that deserves to be remembered. But you kind of have to sort which one fits and which one doesn't. But it was repeated for a reason. So that part of don't discount everything if it's not on .edu. Just, okay, educational shift. When I went to school, it was literally, how can you find what fungus this is? You look it up in a book. In fact, when I went to school as an undergraduate, you brought little note cards like this, and you had to write down the notes, write down the author, the whole 9 yards. In fact, you wrote down the number, then you looked it up elsewhere. And then when I went to graduate school, there was a copier. So I didn't write it down, I just copied it and I highlighted it. And now I would take my phone that's on the charger and just take a picture. But when I copied it by hand, it went through my brain once and I would remember more. So that part of now finding it is not so hard, but sifting it is much harder. Just because it's in print doesn't mean it's true. I hope that is just common knowledge to everyone, but sometimes people need to be reminded. So even when I see it, an old science paper says that macadamia nut quick decline was caused by this fungus. I'm like, nope, that was better data later on said, no, that's not the case. So that was the best information at the time, but the difference, the difference between knowledge and wisdom can be a rhetorical question, but personally, I'm going for wisdom. I encourage all the people you meet in their garden. Knowledge is good, but that doesn't mean you know how to use it and when. Source credibility. I'm going slower on this end. I will go faster on the other end, and you're going to tell me when it's time and there'll be a 20-minute break.
How about 18 minutes about?
Okay.
That's
So this is just telling me how long I've taught. Right in the middle. When I taught biology, we didn't have 3 50-minute lectures. It was in Alaska and the roads are bad. So we had 1 2-hour and 45-minute lecture. Well, the attention span sort of usually left after an hour. It's like, but I have to tell you more information and after a few times I learned that the next class, there was literally going to be 30 minutes of review because just saying it once wasn't catching it. And, I learned over time. So we will have breaks on time and there's good food. The agricultural diagnostics service provided in Hilo that, it is currently $7 for an insect sample, $12 for a disease sample. And Darren's been asked to have it pay for itself. So at those prices, it's non-sustainable. I don't know if someday the prices will go up or whatever, but right now it's a bargain. So, hey, it's $7 for insects, $12 for plant disease. And in some cases, I can find out an answer fast. In some cases, it takes time to culture and things. And in a few cases, I've been allowed to say, could you please bring me a second sample because I can't. So that part about the diagnostician is trying to work with you. This is something where you can help them greatly. How do you get a good sample? So it's only as good as the sample. If somebody says their tree's dying and they bring me a few leaves, well, what if it's a root disease? I can't. And I have had with macadamia nuts, they will go and they will dig some roots and I will look at these roots and I will culture some things and but I looked at, 3 inches of root. Does that really project what's happening in a big tree? I got a small sample. There was one case where I got several samples and 5 out of those 12 samples, I kept coming up with this one fungus and it was known to cause root diseases. Okay. So that part of, I can say, well, I found this many times. I also found this other fungus, Fusarium, which is sometimes a pathogen. It's sometimes just a saprophyte. So it's going to be helping to decay material that has already stopped living. So the saprophytes are not hurting the plant, they're helping. But if conditions change, lighting or something else, they can even, it's called weakly pathogenic. So please, if you can help them to get a representative sample, fresh, some things for rapid delivery, especially bacteria, and then telling us what else is going on in the field. Nowadays, can you email me pictures? It's oh, it's not anything that I was thinking. This picture shows me different, but I can't usually say from a picture alone. There's times where, although with some things like leaf rust, a picture alone is all it takes. There's nothing exactly like that. So you guys, there is actually a handout, all in the details. There's a handout that you could show and give to people. And usually when I was giving a report, I wouldn't tell them everything I knew about this pathogen if I found a pathogen. I would give them a reference and a link that they could go look to it that was more better written and more general application. So this, again, thank you to Brian. We're going to go to the upper right. That is an example of something that Brian liked, because you have healthy and sick. You know what it looks like, and you know, my guess, you know, the leaves look bad, but my guess would be look what's going on in the roots. If you go to the left side, I guess I can, you know, healthy and diseased again, healthy and diseased. Okay, this, it's like $12, and I could waste a lot of time trying to find something there, but I don't know if what I find was actually part of the problem. So good sample with dry paper towel, so it's not wet and going to bacteria. So moist paper towel, plastic bag. When we get plant disease sample, usually Brian would walk right over and get it right away. Well, what happens when they bring it in on Friday afternoon? Like, you know, it has to wait in a refrigerator till Monday, and it's not as good. Encourage people to bring in samples on a Monday or a Tuesday. If something needs to go to Oahu, it's only going to be shipped on Tuesday. And so that part about timing actually even helps. Entire root system. So if you have a little greenhouse plant, this works really well. If you have a tree, obviously it doesn't. But sometimes I was getting greenhouse cucumbers, and there's pythium on the roots that can get into the water. And if it's there, I can usually find it pretty easily. But one time it was the meristem, the apical meristem was dying, and there was a mite that was getting on it, broad mite. All right, I could, as best I remember, and my memory's not what it used to be, but there was a tiny mite that was more problematic in cold weather. And because it was this tiny mite, you couldn't easily see it with a hand lens. But after a while, looking at it under the dissecting scope, that part is, it's not a disease. You know, they paid $12, but it was actually an arthropod, was all that I could find. So, and turf grass. Turf grass is big business. You want the golf course to look pretty, you want lawns to look pretty, but there's a lot going on down there. And actually, as a diagnostician, I've had a lot of fun with turf samples. Look, there's these three things here. And there was this one, it was I don't know if it was a mealybug or something, but Brian knew exactly what it was from the picture. And then I looked it up, and then I knew more too. Now I've forgotten. But that part of, there was more than one thing going on in turf grass. And basically what you're trying to do is shift the balance so the grass is healthy. And if the roots are being eaten by something, the top isn't going to be able to make up for that. So good samples. Diagnosis, healthy versus sick. You want to know what healthy looks like. There's Monono that looks purple a lot. And so that's a healthy plant that looks different than I would think. Development and progression. You know, what do you see first? With coffee leaf rust, you see this light spot. Well, Cercospora looks the same way. But if you wait a few days, coffee leaf rust has these orange spores on the underside only of the leaf, and Cercospora does not. So Cercospora is a fungus that's very common in this, and it will sporulate in the bag, and I can see it under a dissecting scope. So a lot of times I can guess this is Cercospora, but I won't know unless I have access to a microscope. And when I was working 10 hours a week, I didn't even do the insects. I said 10 hours a week, I have to just do these. But the part of if you bring it in, somebody brought a plumeria leaf that had leaf rust. It's like, you don't even have to pay for that. I don't have to look at it under a scope. It's plumeria leaf rust. I'll send you the information about it. But if I have to do something, if I have to do a test or a microscope look, then it's $12. So that part of, how do I be fair with everybody? It's not, I'll look at yours, but not yours. For anybody's, if I have to look under the microscope, then I should count it as a sample. And rapid versus gradual, distribution and spread. A lot of times, if it's in one corner of the field or if it's in the weedy corner, in Alaska, there was a disease called potato scab. And it would be where there had been a burn pile because of the higher pH from the ashes, or where there had been animals fed. And again, this, why is it over here and not elsewhere? Well, these bacteria are doing things in the soil and they just happen to bother the potato as it's expanding. So that part of, is it something that the potato's doing? It's like basically growing in a place that's more susceptible. So the distribution, don't treat the whole field if it's only in one corner. Don't treat the whole greenhouse. History, I'm going to go fast. We still have 8 minutes. I said there's 3 parts and we're getting through the first one very well. Growing conditions, out of place, sunshade. So here they're bringing it in, they're saying what's wrong with it. Well, I have to, and Brian has to, we have to guess if there's too much shade or too much sun. At my house, part of the reason I didn't put the Phalaenopsis outside is I have too much sun. So I knew that you were going to get sun scald on it, but instead I got Colototricum in the house. And darn. Cultural practices. Fertilized, irrigated, sprayed. In Puna, we usually get enough rainfall, but especially this winter, we had like 5 straight days of sun. Well, I would be out watering my plants every day because they were going to look bad. Gardenias will have their leaves turn yellow if they get too dry. So he's like, oh, that must be not enough nitrogen. It's like, no, that could be on some plants, but on the gardenias, they turn yellow and you can just give up on that leaf. You can water it better for the rest of them. But that part of you as a Master Gardener helpline, ask some of these questions. Did you have a cold streak, a sunny streak? How often fertilizer burn if you water it with fertilizer? I just used Osmocote on the orchids because I would let them get too dry and I would get fertilizer burn if I tried to do liquid fertilizer with my watering habits. Not everybody's. Make observations. Proper planting. On the sheet that comes in with a sample, the question, did you apply anything? Because sometimes like horticultural oils in the sun will burn plants. Going back to Alaska, in my experience, little kids, we did soap bubbles. We had great fun on the lawn with soap bubbles, but some of them drifted over and landed on the seedlings that were going to get planted out. And sunlight and soap burned the leaves. We did not do soap bubbles when there were any seedlings out there, but I didn't know the first time. I guess I will tell you this, hopefully you will not do it. I went over to my friend's house in December and I pulled down a philodendron vine with short sleeves and then I cut it into pieces because I was going to throw it over the back fence. Oh, burns. I mean, I visited urgent care twice because the blisters were so big. And that part of, I tell you so that hopefully you'll wear long sleeves and sap of certain plants just this in Alaska too, there was a common plant that it wasn't a problem unless you were out in the sunshine weeding it and then you could get chemical burns. So, to have done this, that was something I never did before.
Wisdom.
And, yeah.
Wisdom.
Well, and the end result was I told the doctor, this can happen because he didn't guess the final outcome. I did because I told him the background. It's like, well, I took 2 types of antibiotics for 10 days and nothing happened. That cortisone was, I don't like to subject cortisone on my skin, but for a short period of time, fix that. So excess or drought, many hosts, if it's on a lot of things, then it's probably abiotic means not living. And that could be lack of sunlight or too much sunlight. So if it's on a whole bunch of different species, then step back and ask more questions. So this may be one more slide and then it will be break time and you'll stretch your legs and everything. Sustainable agriculture, integrated pest management. That's where IPM is the acronym. And literally, I went to school doing this. In California, they have pest scouts and they're paid. In Alaska, we had so few farm fields, there wasn't enough acreage to do integrated pest management. But the first summer I was up there, I was a pest scout. So I went in the field and I tried to count root maggot eggs on and, you know, I was crawling around. I was like, thiazanone might be in order. And so this is another important thing. As a diagnostician, I didn't say you should spray now. I said one option is you could spray this, or you could do, you know, I gave them options. They were in charge. It's their garden. The label is the law. Emphasize that if you choose to use a pesticide, the label is the law and you have to follow it. So that part of you're telling them options, but acknowledging that you're not giving them directions. If they say, oh, what should I do? It's like, well, in the end, the decision is yours, but you could do this, you could do this, you could do this. Very important to say it that way. And if they say, well, should I just sneak this back from, you know, my friend's house in California?
No.
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 There was in Alaska, potatoes being a big crop and disease-free seed viruses and stuff. There was a few occasions where somebody's like, I'm just bringing a potato back. Well, you know, you could bring a virus to the state, you could spread it. It's just, yes.
Potato.
You couldn't do that around me, that was for sure. Cultural control, crop rotation, weed management. What does roguing mean?
Weeding by hand.
Kinda. Kinda. So you guys, good guesses. In a potato field, sometimes I would go rogue for viruses or rogue for bacterial blackleg. I would walk, if I saw a plant that looked bad, I would take it out. And, you know, even do you leave it right there or do you take it out all the way? It depends what it was. Yeah. But roguing is just looking for symptomatic plants and removing those symptomatic plants. It's usually done if you have a big field. But roguing was actually effective for viruses and blackleg because they're distinctive and they're not going to move far. You know, roguing for aphids would be a sad misuse of management. Host plant resistance, yes, please. There is a basil variety from recommended that does better against fusarium. And we have the growing conditions that you grow basil, the susceptible varieties at one time or another are going to get a spore and go down.
Is that red rust stuff that gets all over the leaves of them?
It's more like black at the base,
Oh.
and then the whole thing falls over.
What's making red dots all over every leaf, all over every basil?
I don't know.
Should have brought one.
Yes.
What was it?
Like red spots on basil.
It's on my basil leaf, like a orange, almost like a sting on every leaf, bazillions of them.
You know, that insects can sometimes do things at the right time, but I'm not coming up with something I've already seen that fits that
I
description.
have a bunch of them. You haven't seen them. I just have like them together.
Yeah, there's always, they can email me pictures.
Okay,
That is
I'm
the
gonna do
wonder of technology. But planting resistant varieties, back at Cornell, they have a heritage garden where they plant old varieties. And, you know, they take the plant pathology class there in August, because look at these symptoms, look at these symptoms. You can't make a profit when you're so susceptible, but you also want the diversity. So we have this, you know, don't lose all the genetic diversity, but you can't make a profit on the old ones. So I am past time here. I'm just gonna keep hitting this button and I'll stop till when they all come up. Biological, mechanical, physical, water and temperature, is your African violet happy? Well, if you put cold water on top of the leaves, you get leaf spotting. And it's just from cold water when you watered it. Biorational pesticides, like I was saying, Bavaria is the name of the fungus that is in the pesticide for coffee berry borer, and it happens to work well. I tested something else in Alaska for the white mold I showed you. I saw no difference. So you could pay to spray, but there was no efficacy under those conditions. Good idea. You know, chemical controls, there is a range of things. So it's not for plants, but for cockroaches in my house, bifenthrin is my friend. But I won't use certain things that have a higher toxicity. So think of it, pesticides or no pesticides, you're not seeing the shades of gray. And even a friend of mine, she's like, oh, I'm usually organic, but if I see the squash bug on these guys, then I spray 7 because I won't have any squash if I don't, they kill the adult plant. So asking people to see shades of gray and be judicious and follow the label. And that's where I'm going to stop.
Great. So normally we do questions for about a few minutes.
Okay.
Do you want to do that now or when you get back and save
They're hungry.
your voice a little bit? So, okay, we'll take a 10-minute break and then do questions on the return. How's that sound?
I'm following
Internet?
you.
Yeah.
Let me put it on mute.
Okay.
Hello, can you hear me? Good.
They'll get so tall, they'll get.
Yeah.
All right, we'll get started in a second, folks.
Don't wait for that, they run out.
All right, guys, let's get started. So
I want to thank you for the good food.
Fresh air.
The other islands, I hope you guys have good food too.
So I'll do a few questions over the chat.
I think they may just be looking at the front of the podium right now. The camera's down.
Yeah, okay, I'll get, and you know what, also, I'll adjust
There we
it.
go.
Let's get it a little bit on there.
Okay,
So,
good.
Okay, nicotinoids are listed as a biocontrol. Can you talk about neonicotinoids and the effect on bees?
Details I would be better off looking up. And so nicotinoids come from tobacco.
Oh,
And neo meaning new, so I'm not sure if they're new like it or something else like that. But there were certain kinds that were packaged the size of pollen. This is back from a long time ago. There was something called PenCap-M that was effective on certain plant diseases, but the size of the application was about the size of pollen, so bees would bring it back and they would, it got into hives in a way that it shouldn't. So with neonicotinoids, I don't know if it's the size or if it's just, usually you're trying not to spray when the pollinators are in there. It depends how long the pollination season is or different development stages. So general statements, but specific, I will look it up
Okay,
on
thanks.
a .edu.
Yeah. Okay, would you give some examples of fruit and vegetable plants that are not good to plant near each other as they attract insects or fungi that move from one plant to another? Examples G was interested in is papaya and Cucurbitaceae family, tomato plants, and basil plants. Fruit flies.
So this is where I have less experience with this than probably you do, because when we grew zucchini squash, we couldn't even do cucumbers hardly. And it was just trying to get it enough heat. There wasn't a lot of pests on it. In some cases, they say marigolds around the rim of the garden, but I don't know how that does in the tropics. So I actually don't have good answers, but.
Well, James discussed it a little bit in the chat here with a good, overview generalization that, same family are usually potential sources for sharing diseases and sometimes insect pests.
So
that's-
eggplant pepper.
Yeah, so if you take one thing away from this, which, generally, that's about all I can get out of one thing. It's that same family is usually relevant in terms of disease sharing. And then do we have any questions in person? Just make sure.
so when you're
And then you.
in analyzing soil, do you only analyze soils that have problems, or do you analyze soil with an indication of soil quality, i.e. fungus to bacterial ratios, as well as the protozoa, nematodes and,
So that is an excellent question. When you analyze soil, what do you analyze it for? Do you analyze it if there's a problem or if there's no problem? And then potentially, what do you analyze it for? So Brian worked with soils and he could do nematodes from soils. But if there are soils that you just want to know how much to fertilize, then that is an entirely different process that goes to Manoa and goes through a different thing. So if you want both, you probably have to pay for two different samples.
But at $12, I mean, that's
Well, $12
low price.
is for
Find
disease.
out if your soil has the right ratio of microbacteria.
And actually, we probably can't do
The
that.
price goes up.
Okay,
Yeah.
that's what I was getting.
Just a basic soil samples for nutrients is gonna be, I think
wasn't-
right now, $27.
I wasn't not going to believe that.
So if you talk about that, the amount of work they're gonna have to do for microbial analysis is greater. So, it's gonna be a lot more. You can look at, ADSC has rates, right? So our diagnostic services has the services available and rates on their website. There's also a gentleman and his business, Pete Bond, with Crop Nutrient Solutions on Oahu, who has more services available, and he'll probably have the rates for that. But it's gonna probably start as low as $50 for that. Yeah. Still worth it, sure.
And one thing that sort of goes with that is like, how many soil samples should you take in a field or an area? And basically the answer that I got from college is areas that you will treat differently. If no matter what, you're going to treat the whole field the same way with, you're going to spread the same amount of fertilizer, then one representative soil sample and work hard to get from different places in the field. But if you might do it 2 different ways, 4 different ways, a sample from each area that you would consider treating differently. And then the next question in the back.
Yeah, quick clarification on this slide, under mechanical control, rubber slipper, does that mean to wear rubber shoes that we closed off?
That is a good, Brian wrote it and I would guess that was the case.
Sorry, we couldn't hear all of the questions.
Oh,
Would you
sorry,
mind-
I'm supposed to repeat the question because I'm closer to the microphone. But in the middle, mechanical control, there's row covers, greenhouse, and rubber slipper. And what does the rubber slipper mean? Probably that you can put that in a dip or remove it. And to go with what you just said, I visited Cancun in November for a wedding, and they had greenhouses where they grew some of the food for the restaurants. And they actually had a little depression in the front of each greenhouse that you could and I was like, this is like a tripping hazard, but it's for workers that you would have to step into a dip. So I'm guessing that Brian put that in there, that you disinfect the bottom of your shoes.
Yeah.
No, can I jump in? This is Ty
Yes.
from Kona.
Okay,
was-
Kai.
The rubber slipper it was a humorous addition as an example of mechanical control when you go after one of those 747 cockroaches.
Yeah, I think, but you bring up a good point. If we're talking about from what Roseanne, I keep saying Joanna, Roseanne and Jess are talking about, that would be more like sanitation or exclusion. So it would probably go up into cultural control.
Okay.
Which is a good point. Right.
So you could also put fly swatter if I'm hearing Ty right. So this is actually the last slide on the first of 3 topics. So it was actually good timing. But was there any other questions?
Internet?
And like I said, I have a more informal style and I don't mind stopping and starting. And I never know exactly what part of Chuck's story might come out when I give these. So I'm not rigid in my presentation.
Let me just. Give it up.
I hope you can tell I have enthusiasm and I enjoy talking in front of people on this kind of subject. because usually people don't really want to hear about plants dying. And while he's checking, there was a garden fair in Palmer and they asked me to come and I'm like, well, I could talk about lake blight. Well, it's a gorgeous day and I'm in a barn, you know, and I think 2 people came in. I sat there for 3 hours. I'm like, well, I would rather be outside too. But that,
We're good.
okay. So we're trying to go for the next one. So if it's yeah, I could.
And then, so just now you can go.
Okay. So usually, especially I would recommend for you guys, one of the first places to look is on the UH website and the little publications. So I don't know if it's not as easy to search as I would like, but if you keep trying a few different keywords, you can either try the host or the common name of the disease problem. And sometimes there's a little 2-pager or 4-pager or something that has a lot of information that you can give that link to somebody or print it out or whatever. It's better not to think that what I say, it's like what I read is what I tend to believe more. So plant pathology, the diseases, as I said, I've studied this a long time. There's a challenge in summarizing it, but I'll go for it. And this is the proper definition, but I'm not even going to read the whole thing. I am going to talk story and say that in a molecular plant pathology class where they're talking about.
Sorry, we're only seeing you in the podium. We're not seeing the slides for some reason.
But we're not showing. Good, pointing it out.
Oh, we can see the slides on Oahu.
Yeah, I can see the slides too.
This is just taking a little while.
I'll play around with it on the side.
I see the corner.
All right, we fixed it over here. Sorry about that.
No worries.
So the study of organisms and environmental conditions that cause disease in plants, the mechanisms and interactions. But there was a test in this class that I took, and these molecular people, it was like, what is plant disease? Well, I'm a big picture person and I had a really fun answer. And the professor said, you know, there was only one person that knew how it's basically a disease is outside the normal range of health. So that part, there is a range, but when you go outside it, it's kind of like, when do you call it a disease versus just a condition? And that part where there's shades of gray, but the molecular people that I work with in graduate school had James Watson, you know, one of the 2 that discovered DNA. He had an attitude the size of Texas or Paz or whatever. And I do, and it's, in some cases, the people that did molecular research use that as a role model. And so sometimes it was hard because I would hear, oh, it's too complicated for you to understand. I can't even explain it. And I would get very upset because my role model is like Richard Feynman, who's a physicist. He's like, if you're good at your subject, you can explain it to a kindergartner, you can explain it to the public, you can explain it to your peers with different words at every level. And that is what I use for a role model. So I'm very sad that some molecular people, there was a trend of saying this is too difficult for most people. Therefore, now people are afraid of GMOs because, you know, it's not explained well. It can be explained, and there are shades of gray, and there's, you know, reasons for stuff. So I advise that Richard Feynman is a better role model than Jim Watson, in my opinion. And the disease triangle, usually it's lines, but here we have circles, and the center is disease. If you cut off a circle or a corner of the triangle, you will not have that condition that's outside the normal range of health. So if you have a resistant host instead of a susceptible one, you can stop it from happening. The pathogen, if it's excluded, you can stop it from happening. But the conducive environment is often easier to manipulate. Some of those, you have a host and the pathogen's already here. With fungi, air circulation is this huge conducive environment. If you have better air circulation, you have less disease. And it's something that is, not harmful to the environment, but that part of, less weeds, putting a fan in the greenhouse, so that conducive environment, also free water on the leaves. If you're having a bacterial problem, the bacteria can't fly. They don't drift nearly as much like fungal spores. So they're going to spread in the film of water. So if you don't have water splashes, if you don't have a place under the pots where water goes from one pot to another, you're going to minimize the spread and likelihood of having a bacterial disease. And as I said, when you start thinking in these terms, paranoia is not a bad thing. Was it on my feet? Clean them off. One more silly story. When I was in graduate school teaching plant pathology as a TA, teacher assistant, and we had a greenhouse of plants that were having symptoms that we could show to the students. And every time you went in there, you had to put your hands in soapy water. So this wasn't feet, but you have to do this so you don't spread viruses. You put your hands, you never smoke in a greenhouse with tomatoes. It's on your hands, the back of the mosaic viruses go back and forth. But my daughter came in and a little later we're home and she caught a cold and she's like, but I didn't touch the plants. Like, yes, cold virus, plant virus, but they're not the same. And I was glad that she was making these associations and then to explain that there's a lot of viruses and many of them are very, very specific. And some of them are just nasty, like corona. Yeah. I realize that the children growing up in this, they will never have the same worldview that I have of not having this till I was in my late ages, and now I'm happy that when I go on an airplane, I can have humidified air by wearing a mask, but that part of people are becoming afraid to touch. And so there's a time and a place to minimize the risk, because certainly, but with plants in a greenhouse or in your farm field, mechanical spread is a big way for certain viruses. And the conducive environment for viruses is, basically you want to prevent spreading it around. One more, even though it might not go with this, is in Alaska, save seed potatoes to plant the next year. You're having carbon copies of the parents. But okay, you sell the big potatoes and you keep the small ones to plant next year. Well, the small ones came off the virus-infected plants. So, and then the yield is down because there's a virus in the potatoes. So I'm saying this because sweet potatoes and taro are the same method of having clones of the parents. And so you want to have clones that show no virus symptoms, and tissue culture sometimes can increase them and all of that. But the part of, excluding the pathogen by starting with a clone that's virus free. This, Brian is right. We've talked about it. Abiotic means not living. And we tend to think, oh, where's the pathogen? What caused it? But too little sunlight, too much sunlight, lack of water, too much water, nutrient deficiencies. There was a certain palm that it would show a wrinkle on the palm if it had this nutrient deficiency partway through. And now I can't remember which one it was. Ty might remember better, but there is on the website for UH, an example of a picture that this palm tree has a nutrient deficiency that usually comes during times of high rainfall. It's washing things away. So it's not even, is it the nutrient or the conditions where the root can take it up? Temperatures. Now you guys probably haven't seen as much, but what's wrong with this? Well, it looks like frost damage. And in a lot of places, early season, fruit trees, frost damage will damage the buds and they won't set fruit. But lack of oxygen. When I'm not thinking the above parts of the plant, but if you have waterlogged roots, the roots also have to have respiration and they can have a lack of oxygen if there's too much water. Pesticide toxicities. Just saying, I did variety trials of lettuce and cabbage in places, and it took a lot of work to grow 20 varieties. And, we individually planted the seeds and seedlings, we planted them out. And in this one field, they had potatoes before, they had cool temperatures. And I come out and a third of the plants are just flat, they died. Because the cool temperatures in Alaska meant there was a carryover from the year before. And of course, the grower didn't know when they put me in that place that might be the case. But, it was literally a season's worth of data lost. So sometimes it happens, the label is the law. Pretty much if you follow the label, that's going to be a minimized risk. But sometimes somebody could describe drift from their neighbors or the roadside application. And, you have to consider the possibility that might be true, but also that it might just be a red herring. I can't answer those questions. Air pollution, we saw too much or too little light. Going back, especially this center one, there was a coffee grower that said, unless he pulls up the little coffee plant, he won't grow a coffee plant that was in a container because it's probably not going to set a good taproot. And in the long run, the life of that coffee plant will be less. Now, I know that I have had plant roots that look like that because I waited too long.
We have a question online.
Okay,
Can wind cause abiotic diseases?
I would say yes. But again, a condition outside the normal range of health. So is this? Was there another? Okay. Sometimes it could be just physical damage that doesn't persist, and that would be like injury rather than disease.
Okay.
But there are places where it's very windy, and so there's this repeated wind. Hardening off plants. That there is my niece in the Midwest, and she buys a zucchini plant. Now, some of these, they've actually had fans blowing hard so that they will be ready to be outside. But if they get a fast wind, that zucchini will start windmilling till it ruins this thing and the plant falls over and dies. Again, that's technically probably injury, but it's a problem. The other bit to go along with wind is my husband would start plants in the house, and then he finally just said, I'm going to start them outside because that they actually grow faster if they've grown outside and been toughened up by being there. So that part of when do you start it, how do you toughen it up? But if you try to cut those circular roots, maybe, but maybe not. If it's an evergreen plant like a pine tree in New England, you just throw that plant out. And actually there was a plant from Home Depot that had grown too big, so they cut off the top and they sold the other plants for $5, and I bought 6 of them. Well, they were so root bound that they eventually all 6 died, but they before they died, they went through this they turned yellow because they couldn't get enough nutrients, and they looked really festive for about 2 months before they died. So I'm thinking $5, it's okay.
Yeah.
But on the left side here, planting too deep, like that's not true for hibiscus. That would be true for a strawberry. That would be true for some things. I'm actually not quite sure there, but what do we see happening in this bottom right? Think Fort Lauderdale. Yes, flooding. So less in lava, but in some places in New York State, they actually put tile under the field to drain those waterlogged places. Planting too deep. Again, there's only certain plants that it matters more whether it's root or shoot. There's plenty of plants around here that you can take a cutting and put it in. But again, this mulching part, if the mulch has a lot of activity going on from bacteria and fungi and arthropods and other things, that can damage tender bark. And, so it wasn't actually that it was putting it in close proximity to something that damaged the bark. So the recommended planting depth is very important for certain plants and not others. Disease types. This place on the right here is taken from the plant pathology book by George Agrios that I used it as an undergraduate. I used it 10 years. It's a classic. And I like that it's showing you the big picture of the differences in size. So we have moved on to things that are living that cause infection. Although we have viruses here and viroids, and the question, it's like a comprehensive exam question. Are viruses alive? Well, they're a separate entity, but they can't live without another living cell. And basically you explain that it's a gray area for these reasons, and you have given a competent answer. So that black and white thinking is often not helpful in biology. There's a lot of gray areas. When I taught biology, I said there's an exception to every rule you can think of. So the size, fungi and water molds. Water molds are actually in the kingdom protists. They have cell walls that are closer to plants than fungi have chitin in their cell walls. Water molds have cellulose like plants. And chitin is actually in the insect exoskeleton. So this major difference at cellular levels. The whole big thing here, this is the plant cell membrane and then the cell wall. And then you have a nucleus. So as a biology teacher, just the cell is the basic unit of life. What makes up a cell? It has to have a plasma membrane and DNA. If you're a bacteria, you don't have a nucleus. You just have DNA floating around inside that plasma membrane. And you have sugars usually in the wall outside. So there's all of these exceptions and interesting things to study. But a plant will normally have a defined nucleus. It has a bunch of organelles, and it has energy from sunlight, and other things are trying to harvest that energy besides us. So the head of a nematode. So this is a little roundworm we'll talk about, sticks its head in, slurps it up like a Slurpee. Bacteria are mycoplasms. So mycoplasms are a little bit smaller than bacteria. They're usually, they used to study what they look like growing in a colony, but now they would actually probably extract DNA and say, what are the patterns in the DNA sequences? And viruses and viroids, very small. I can't see these under the microscope in the lab, but they have dipstick tests that you can do for a lot of viruses. In fact, they have them made so that if you have orchids, you could buy your own set of 100 and test those orchids if you so wished. So we can do it here, but if it costs $12 and you buy in bulk and it only costs $8, we actually mentioned that you could buy this and test yourself if you're doing a whole bunch and you know what you're testing for. Protozoa they have single cells or more than one cells, but very simple organization, not a lot of specialized tissues. And there is one that I don't recognize, but technically water molds usually fall under the protozoa kingdom unless they've divided it up. So there's no lack of things to study in biology. And I'm telling you what I have read about, but there could be things that are newer that are being written about. So keeping up with the latest is a wonderful thing to do. And it can also be the starting point of a discussion. So if there's a person discussing, I thought that fungi were small plants, you can say, yes, that's what people used to think. And, at a certain time, they discovered these differences, and let it be the start of a discussion. If they say, I don't know about GMOs, you can say, well, there are some benefits, there are some disadvantages. Let's make a list of the pros and cons, and then you can balance those, because insulin is an example. Most of insulin these days, if I understand correctly, comes from GMO cultures in vats, in laboratories. It used to come from pigs. You're trying to get this stuff extracted from pig carcasses in a long time ago, and it's not as clean and not as specific. So in the case of medicines, I'm liking this, that insulin is produced better in that way. When there's soybeans that have things so that it's resistant to an herbicide, it's like, that's not really benefiting me, that's benefiting the bottom line of Monsanto. And so that part of, the pros and cons are not as favorable to people as they are to corporations in some of these cases. And if you can let it be a discussion, because each person, to respectfully talk about different opinions, and that science is continually asking questions and getting new answers. Yeah, please, as a scientist, the more education you can bring, and it lets people be less defensive if sometimes you say, well, why do you think that way? Or, can you give me an example of that? If you ask it as a question, that people's defenses aren't as big. So asking questions rather than, tell me about this, what did you see first? Or why do you think that? It's very helpful in my experience. And if I worked as an ag extension agent, if I go to a farmer's field and I say, oh, this is your problem, like, what? You didn't talk about the weather. You didn't admire my plants. I have no credibility. So you really do have to do a little talk story first that is respectful, and just saying, you
Gonna have lunch.
luxury yachts, is it that the Supreme Court can do luxury yacht trips? We can't. So this looks remarkably familiar. Somebody brought,
It's a magnolia leaf.
magnolia leaf. This was because I had way too many words from Brian. I don't imagine you guys can see that very well, but you can see on the screen, this was a picture I took. I'm guessing guava. Anybody besides Russell that knows what it might be?
Come on.
Come on.
All mine?
I can tell you that I never saw it in Alaska ever.
You got this.
So then it has to do with moisture then.
Yeah. And
Come on, guys.
Sorry.
Too much rain.
Some kind of fungus.
It's raining heat.
It looks like a fungus, you know, but it's a parasitic algae.
Parasitic
And it was just like, whoa, Cephalurus. There is a 2-pager, 4-pager on the website that talks about this. And usually, you know, increasing the air circulation, it's usually not going to be big enough to require treatment. But it's definitely showy, and people want to know, what is this? What should I do? So if it's on the lower branches and the upper branches are doing fine, the tree can do fine.
That's probably more like it.
Yeah. And, you know, if you need to, it's leaf moisture that allows it to do this. And there's actually 2 species. One is only on the upper side, the other's on the upper and lower side, but a parasitic algae. So that would be a protozoan rather than bacteria or a fungus. And it's showy. It's what I can say. Cephalurus is what I remember.
So visually now, you just look at it and you know immediately that that's what it is. But initially, did you have to examine it under a microscope to make that determination?
So good question. He's got it right on. Initially, I had to look under a microscope and read about it. But Brian, he takes one look at, oh, this is,
yeah.
I'm like, oh, okay. And then I read the book, and now if somebody brought this to the front window, I could say, you don't have to pay me $12 because I could tell you what it is without looking at it under a microscope. If they come when I'm not at the front window, they would pay $12 and I would still give them the same information.
It's not contagious though.
Um,
I mean, should everyone in the class stay away from me?
no,
Okay.
because it sort of sloughs off a little bit. It doesn't fly through the air as much. Water splashes.
And it's
Okay.
already ubiquitous across
Okay,
all of East Hawaii. So, yeah, I
So
mean, it's everywhere.
It's more conditions,
okay.
you know, a little drier leaf surface, and you won't have this parasitic algae.
Go out there with a blow dry.
So somebody suggested blow drying the leaves might help, which probably would, but it'd be lower.
Serious.
If you had to do the cost benefit ratio for that. So now we are coming close to the next hour, but nematodes, viruses, and bacteria are really fast. They're not, so I can probably do 1, 2, maybe even 3 quickly. And then I can talk about fungi till the cows come home. But I won't. I will only talk about it in a given length of time. So nematode diseases, the picture shown here is looking like a golf course or an athletic field. And, you know, the spots don't look good. They're not as good for traction. And, you know, nematodes, it's kind of indistinct. And I can guess, well, maybe, but that's one of the ones where I would pay $12 and Gabriella would have a lot of work. Very small microscopic roundworms. The people that study them say they're the most abundant and diverse. I kind of, you know, think ants and insects personally, but Brian had this. Middle, there we have indistinct and usually mimic nutrient deficiency. And they're actually very common in the tropics.
Okay. So
So that part of, you know, if the plant is just sort of going down and in generally it should be doing better, you know, and the roots, you know, check what the roots look like. Almost all plants, soil assays are necessary, but no fun. Let me tell you, that's why I didn't do them. 10 hours a week, but they are available now, and especially if you have a larger area or commercial growers, it matters what kind before you apply a pesticide for that kind. So if it's big enough, you want to pay for the identification. You know, if it's small enough. And it is similar to bacteria and fungi that most nematodes are free living in the soil and they are not damaging plants. If they don't have a stylet, they're not damaging plants. But that small percentage that do have stylets some of them are very specific, some of them are not. We happen to have a root knot nematode in Kona that actually is named Meloidogyne coniensis.
Oh.
You know, it is specific, and the favorite type of coffee trees will die in 10 years if it gets a high nematode infection. So what they have to do is graft this preferred coffee variety onto resistant rootstock. So the roots are affected, but not as badly. And it's a lot of work to graft coffee seedlings. So that part of, you know, it may not be a problem for a lot of things, but in particular cases for coffee growers here on this island. And so it's in Kona. I don't know that it's in Ka'u as much. So do you want to bring seedlings from Kona to Ka'u? You might want to think twice about that, you know, for exclusionary purposes, because once it's in the soil, you know, it will spread when you spread soil by footprints and tires and stuff. So it is a common problem here. It's hard to say for certain, and if it matters, you know, you can also ask if they were going to consider treating the soil, or just, you know, start with new stock in a different place. Just so you've heard it once, endoparasitic is inside. Ectoparasitic is you have these little cysts sticking out of the roots. The entire body is outside the roots. And, you know, if you read the literature, you'll see these terms. You don't have to remember them. You'll find them if you ever need to. Here, Meloidogyne incognita. Guess you can't recognize it very fast. But, but on the roots, you think that these roots are going to pick up nutrients as well. So this is healthy, this is diseased. There is a Meloidogyne chitwoodi that is in potatoes in the Northwest.
Are there CTAR publications specific to nematodes and also specific
Yes.
to nematodes on Oahu? Or is it just Hawaii specific?
So my best guess is more like Hawaii, but I would look up because whatever I have is at least a year old at this point.
Okay. Thanks.
They used to use methyl bromide to gas the soil for nematodes. Are they still doing that?
So methyl bromide was used to control nematodes, and it basically was a biocide that, and it's a control measure. I'm glad that
If they don't do that anymore?
it is too dangerous.
Okay, it was for ginger, I do believe.
So that's now where you have raised pots.
Okay.
Pots and raised beds is a better.
These are farmers, large spreads.
Yep.
And tarping
And I have seen that in other places and in California and stuff. So this, it's like the 50s method is just sterilize the soil, forgetting, you know, well, what's going to be the first thing to colonize it? It's often not the things that are helpful.
Yes.
So that part about killing everything is out of favor for very good reasons.
Okay. Thank you.
You're welcome.
Of the endo or exo?
Well, here is
So
it.
is that an example of an endo or an exo nematode, just for the?
Yeah. Endo or exo, this is endo and this is exo. And again, they're showing here how that's inside the roots and stained.
Okay.
But this, the coffee plants in Kona, you can literally just pull them out of the ground and they start to fall over. So if your plants, you can pull up and you see roots that look like this, you can guess that you don't want to plant that in that area anymore.
Roseanne, may I ask a question about the symptoms that you're looking for in the upper foliage to even suspect those nematodes? Like you said nutrient deficiency, but can you say anything more about what it would look like?
Well, failure to thrive. So like they said, it's a general sort of thing. You know, the plant should be doing better. It's getting enough water. House plants, a lot of times, if they look wilted, like, well, then I must need to water them more. Well, then they have root rot because the roots are too wet and you water them more.
Yeah.
You know, you guys can be such helpful educators. More houseplants die of root rot than a lot of other things from overwatering. So I guess not much to say what's the symptoms, but actually we have an ARS person that works up the hill. She's a nematologist. So Maloidogyne conensis is one of the things she works with, but she has publications with pictures. So
I think you look at it like this, right? You've got a plant that's showing symptoms of that show that the roots are damaged, right? So it's either chlorosis or nutrient deficiencies or it's wilt. You start to look at the causes. It could be abiotic, where it's too little, too much water. It could be a nutrient deficiency. It could be a fungal pathogen, or it could be nematodes, or a number of other things. But you have to go down the route and try to find the answer. So a lot of these symptoms can look the same for different diseases, or look similar, right? So that's
this
important to consider.
is the last slide before break because I believe it's the last one on nematodes. We'll come back and we'll look at viruses. And I want to see this Aploencoides frigeriae. And I was told that when I first did this talk 2 years ago that most of what they remembered was, how do you say that word? How do you say scientific words? And my answer, and you should practice this, just say it with confidence. Caribbean, tomato, Aploencoides frigeriae. There will come to a fungus called Colototricum. Colototricum is how I heard it on the East Coast. I heard it up here, but we had some people from Puerto Rico, Coletotricum, and she gave her whole talk that way. With confidence, there multiple pronunciations are okay.
Okay.
So this is a nematode on the upper part of the plants. I have not seen it. I would like to see, it's on my bucket list, but I don't want to see it in my garden. So I probably should just tell Gabriella, if she ever sees this, call me. So this is what comes after break. Is that good?
I'm seeing,
Then
I just had a orchid with that. Yeah.
Quick.
I couldn't figure it out, and ended up being the voice.
Okay, so 10 minutes. Yeah.
okay. You said don't show me.
We're gonna get started again. I think we'll do questions. All right, folks, I don't want to do my voice.
Making use its outdoor voice.
Can't today. Any questions? Online or from the other islands?
So if there's any questions, they could be now or they could be at the end. We have viral, bacterial, and fungal diseases. And fungal diseases is a fit to fill the amount of time I have.
Okay.
All right, there's no questions.
Okay, viral diseases. There is a smaller number than fungal diseases, but they are outsized in importance. Because basically, if a plant has a virus, what do?
Chop it.
You chop it, you throw it away. There's no easy, worthwhile way to rehabilitate it. If you have a huge, beautiful citrus tree and aphids came by and gave it citrus tristeza virus, you can, because it's in the area, you can leave it to be a source of more virus for other plants, or you can chop it down. The saddest I saw was a school garden, and they had a big stand of bananas, and they were having the kids chop them down because they had bunchy top. You can't keep a few, it has spread through the whole thing. And orchids, prize orchids, if it has a virus, keep it and it will spread to other things, either mechanically or by insects or arthropod vectors. So, you know where we're at, and I'm back to the press the carrot before. I love that Brian did this for me. So we have papaya, I believe papaya ring spot. There is a website with all of Scott Nelson's pictures. They papered on Flickr, and there's like 200 shots of banana bunchy top. So it was, but that part of these little guys, once you've seen it a few times and you've confirmed it's like, now I would be confident enough in my guess. But if you're asking somebody to chop it down, you might want to pay $12 to have it tested. There's cucumber mosaic virus is actually even found on some weeds around here. Papaya ring spot virus was threatening to wipe out the industry because the yields go down so much and it spreads. Some of them are mechanical spread, like the ones that I work with in Alaska on potatoes. It's literally when you cultivate to kill weeds, you're spreading viruses from plant to plant with plant sap. So that part, sometimes it's people, but then sometimes it's aphids or some other piercing, sucking mouth part of insects. So, and those are harder to control. So when it's spread by aphids, as especially in the case of banana. So I don't see my mouse. There it goes. I have to press harder. But again, the details of each one of these are pretty well known and you can check in the literature for them. But if you start to see this funny modeling kind of thing, start to suspect a virus. I mean, there's a few other things that might make it look that way. This puckering, misshapenness, little necrotic dead spots. So.
Question online.
Okay.
So for the banana bunchy top,
Yes.
Does cutting it down cause the aphids to move, and what are the ramifications of that?
Very astute question. That means a very good question. The recommendation is don't cut it until you spray the aphids, because they will move to the next one and they will carry it as they go.
Spray.
And, you know, there's recommendations. I can't do them off the top of my head, but we actually have a little trifold, and Hawaii County had a grant from the Hawaii government that paid for 75 tests for banana bunchy top. But we asked people to sign a little piece of paper saying if it's positive, you'll follow the best management practices, which said you spray it and cut it down. But yes, if you cut it down without spraying, then your neighbors will not thank you. So vectoring is carrying the disease. Vectoring the diseases, we have persistent and non-persistent. I like non-persistent better because that's whatever's stuck on the straw. You know, there's a little bit on the mouth parts. So, you know, the next 2 or 3 places it feeds, and then it's done with the virus. But there are other cases where the aphids get it in their gut and they are uruliferous for the rest of their lives. So in that case, the aphid is a problem for a lot longer. Now, off the top of my head, I don't remember which viruses permanently infect aphids and which viruses are just mechanical, non-persistent. But it matters when you talk to people because aphid control becomes way more important. And, you know, in a farm field, how far does it spread? That part of, you know, the one case where we talked at break about marigolds, and they said that there's a CTAR publications that they studied it. The numbers that they got from that study said some varieties of marigolds have some effect on some nematodes, which means it's hit or miss. But again, you can also plant border crops around virus stuff and sometimes have a non-persistent aphid lose the virus before it gets inside. You know, so that there are things where borders of different kinds of plants work fairly well. And you can, this was one that I added. This was from a sample. It was from an orchid. So this little dipstick here is for Cymbidium mosaic virus and what's RSV?
Odontoglossum ring spot virus.
And so this is 2 for the price of 1.
Wait.
Over here, it shows you if you have this pattern, you have a positive for both viruses. If you have this line, you have a positive for one virus. This line is positive for the other virus. So what does this person have? The one that begins with a C or the one that begins with an O? Yep, I agree. It's Cymbidium mosaic virus. You know, it still has a virus. You still probably want to get rid of the plant, but you only had one. This is a measured amount of plant. I actually have to weigh the amount, usually picking symptomatic material or whatever they tell you. So that's, again, if I pick the wrong place on the plant, I could come up with a negative. You pick the place where it's most likely to be positive. Usually you would grind it up, you know, you could do it with an eraser, mortar, pestle. We have a drill press. Brian said, you know, his fingers were past that, and I really had fun with that. I got to do it. So this helps break it up, and 15 minutes you have an answer. So if somebody brings in a sample and they're thinking virus, I can give them an answer the same day. It's kind of fun that the price of the chemicals, though, is nearly the price of the sample. So it's like my labor is free, or, you know, somebody's subsidizing. Done with viruses, but that part about they're very important, and ask people to think twice about, you know, removing the chance of spreading. Bacterial diseases. Again, these are less common, and they're usually associated with water moving the bacteria from one place to another. But in some cases, they're a big problem. This being the big problem. So bacterial wilt, you know, you see that the plant on the left is had it. And that's because the bacteria have clogged up the waterways. So you can water it all you want, you're just going to spread the bacteria to underneath the bench. But this, you know, I cringe when I hear Ralstonia solanacearum because you don't want this. And basically, if one plant has this, then you can mechanically move it to other places. So usually if it's in one place on your farm, you know, it will spread as you walk in that place and touch that stuff. So on the other hand, if you grow something up on a bench and you clean the pots and you use different soil, you can use that space, but it costs more to grow it on a bench, and it takes constant vigilance so that things don't get from the floor back up to the pots. So this being one that is fairly common up here, and you know, sanitation, exclusion, to try not to bring it into your little garden is my best bit of advice. So more fun bacterial names: Erwinia chrysanthemi, and they may have changed Erwinia to a different name based on naming stuff. Xanthomonas, on Russell's favorite, anthuriums, Pseudomonas. That's bothering lettuce, I think. And again, it looks water soaked. So it's going to spread by water splashes and water touches and stuff like that. Down here on the bottom, Burkholdia. So there was a person named Burkholder who did a whole lot of work in that stuff. But I like bacterial speck and bacterial spot. You would guess that the size of the lesion is probably what's different in those two for those common names. And then here being a place where, you know, death in this, it's probably a wet spot in the field was Ralstonia. But tomatoes get a lot, orchids get some. And what I saw is you have this, it's a weird symptoms, like up here would be the water droplet, and when you have high humidity, the little bit of water gets stuck on the very tip. Well, a little bacteria gets in it and it works backward, is my best guess. So most other things aren't going to start from the tip and go backwards like this. And if you're very careful, you know, it's possible to just cut off that leaf, but you don't want to put it any place where it's going to splash onto other things. And bacteria cell walls are broken down by soap. So just washing your hands with soap is a very helpful thing around bacteria, viruses, you know, plain old soap. It doesn't have to have the other stuff added. And one more of these, the streaming out of the bottom of the stem is literally a bunch of bacterial cells. So if you splash this water on other stuff, it would be bad. There was another time where I had a really quick diagnosis. It was a plant and they asked me if it was bacterial on the leaves. And I can't say what the bacteria was, but it was the right shape and the regular small size, and they're in abundance. So I'm saying, yes, this is probably the bacterial disease you asked about. Now, you know, in that case, I think it was a commercial planting, you know, you could take steps based on what they have available for that bacteria, which is usually coppers. We made it! We're on fungal diseases. Sorry, I said I had to leave Alaska to take a mycology class, the study of fungi. And yes, I like this stuff. At the same time, you know, there doesn't need to be that many people that study it. And you're going to look at ways to minimize it that usually never get down to to chemical control. It's going to be changing the environment or excluding or using a resistant host. But some of the words that you come in, fungi grow in little strands called hypha. Hypha is a single strand, hyphae is many little single strands. So on the left here is a photograph I took, I think it's from turf grass, and all these little strands would be an example of hyphae. Now, these little other things, it's called, the fungus is called curvularia, and it can bother the roots of turf grass. But these little things that look like croissants to me are the spores. And the spores can wash with water. If they're above ground, they can sometimes blow on the wind. But the little spores can move places faster. Most of these spores are going to be exact replicas of the parent. There are a few things like that come out of mushrooms or disc fungi that have some genetic recombination, like flowers and seeds. And then mycelium is when you can see it with your naked eye. You don't have to use a microscope that you see this pile. It's actually a sign of disease when it's the actual pathogen growing there. Whereas the water-soaked area, like over this would be a symptom. A symptom is a plant reaction. A sign is an actual visual of the organism if it's a biotic disease that's causing it. So as a diagnostician, I look for both symptoms and signs. With viruses, I'm only going to see symptoms. With fungi like this, the color of this is Colototricum on papaya. Colototricum, Colototricum is very common up here. And if it's allowed to develop, which you may not want to, let it, but you'll see this, it's like peach or salmon colored. And the spores are plain and little, and it's not easy for me to say that these spores are Colototricum, but the color of the mycelium is more diagnostic. And part of it's luck of the draw. If it lands in a nice wet area, especially with overripe tissue, then it might develop. I have a crown plant that keeps growing sprouts, and then Colototricum knocks them back, and I put some more sprouts, and I'm never going to have enough to feed monarch butterflies. I'm sad about that. So many, again, Brian picked this. How do you choose? You know, spots on a rose leaf. This is worldwide botrytis, gray mold. And while it's worldwide, it is a weak pathogen, meaning only when conditions are really good, you just increase air circulation. Usually it's never helpful to spray for gray mold, but that's a problem. You're not going to eat those. And the powdery stuff that blows off is looking for any other place that is a little bit overripe, ready to go. So this is not a fungus. What is this? Nematodes, thank you. And this is also that aerial nematode picture. So this is a fungus rather than on banana leaf. And poinsettia with powdery mildew. Now, I'm not even, plumeria, I'm guessing that's plumeria rust. And to me, that looks like thrips on a pepper. So as a diagnostician, I start to like, well, my best guess is, and then go put some background into it. If it's this, then I should be able to find thrips or thrip droppings or other things from their raspy mouth parts. You know, that part of, I can make a guess, but I usually want to follow up on that. And of course, I'm wrong sometimes, you know, but sometimes I'm not. So don't feel bad.
Where can somebody find the latest treatments for bacterial wilt on the CTAR website?
So saying bacterial wilt in the publications is a good start.
Yeah.
sometimes I also would go to the florida.edu sites.
Yeah.
And see what they say, Florida, California. So if CTAR doesn't have it,
Yeah.
because it has to be updated, try another tropical state.
Yeah, I tend to say, if I want it from CTAR, I'll write the disease or the issue and I'll write CTAR after that. And if I want to find it from somewhere else, I'll write just extension service or extension, and it'll send me normally to other extension.
Well,
That's through Google.
if you go to the UH website and then you go to cooperative extension and then you go to publications, so then you can just, there's a search bar for CTAR. So I'm not getting the whole World Wide Web.
I personally think that search engine though is really bad.
It is.
I
It
tend
is
to use
really
Google
bad.
instead of, and I still find the CTAR pubs, but I do it through Google because I don't think it's a very good search engine.
For some reason, it doesn't pick up all the words and yeah, I would agree. Sometimes I just started looking at the whole publication list or look at the host plant. So I'm sorry that that's the case, but the website on UH may not be as up to date as some California and Florida. So
Can I ask if it's fungus, what do you then suggest? Cutting off the leaves and then removing them far from the plant?
so it depends what kind of fungus.
Okay.
So on the gray mold,
On the banana.
on the banana, I would actually look on the CTAR website because we actually have a picture of that and they have more information. So I don't want to say when I know you could find better.
Okay.
And we could see that this afternoon. Filamentous organisms, they grow by hyphae. Now mushrooms are the fruiting bodies of one group. Discomycetes are the fruiting bodies of another group. And then there are some fungi that they don't know what the fruiting body is, but they still cause disease. So you can have a name for the fungus that doesn't have a known fruiting body. You can have a name used, and then they use DNA and they find a different rearrangement and they reclassify them. So fungi, you're looking at multiple names and you kind of have to be a sleuth to see what year it was written in as to what name they might be using. And going here, 85% are caused by fungi. 100,000 is the estimate when Brian wrote this, and only 8% of those cause diseases. So what do the rest of those 92,000 fungal species do? Beneficial or saprophytic? Saprophyte is an organism that lives on dead tissue. So when the leaves fall off the trees, the saprophytes decompose them into something that's better for soil, into smaller pieces. And when I talked earlier about the kingdoms on this planet, plants make their food. Animals ingest their food. It only works if you put it in the mouth. If you put it in the ear, it won't digest. So the protists can do either of those. The bacteria can do a lot of different things, but fungi do not make their own food. They do not ingest their food, but they actually, with this long skinny body of hyphae and mycelium, they absorb their food. So they put their digestive juices, and it's like living in a stomach, basically. They're decomposing outside their body. And decomposers are really helpful in ecology. But as I said, in the garden, they harvested it before I did, and I'm mad about it. Okay, more pictures. Cercospora leaf spot. I see this on tea plants a lot. I brought it in for Brian, and he's like, if you fertilize it, the Cercospora doesn't get the upper end of the balance. So he didn't even ever recommend trying to treat the fungus. It's improve the health of the plant with fertilizer.
That looks like what's on my basil.
Well, and if, as a diagnostician, if I put this in a moist moist chamber, basically put it in a Ziploc bag. Then there'll be these long skinny spores on there that are diagnostic characteristic of this. So that's something that I could do in the lab that I can't do at home. But just having done it in the lab once, I can say my best guess. Over here is plumeria with rust. And again, there's a great CTAR pub. Usually it's like the parasitic algae. A plant can tolerate that for a little while. So it looks bad, but actually just let it go. It'll be okay in most cases. Brian had it, I left it in, you don't have to remember it, but talking about fungus breaking all the rules, I said a cell is the basic unit of life. It has DNA and a plasma membrane. So these over here, we have a cell with a nucleus. A septa is dividing a cross wall if you want. You know, that septum, a pore to allow things to go back and forth more easily. So they call this a septate hypha. And that fits the biologic concept pretty well. Over here, cenocytic hypha. What don't you see? No septa, no cross walls. How do you tell what's a cell? They're just nuclei floating all around. Well, it works for them, but as a biologist, you know, I don't like this. I can't tell where a cell starts and ends, you know, and there's multi-nuclei in there. And the fungus that I work with, it's called Rhizoctonia, and it's multi-nucleate. And then there's some binucleate ones. Like, what do you mean? It has more than one nucleus. They can have different information in each one. It's like they're breaking the rules if you think of them as rules, but actually they're showing the variation. And then in biology, if you look hard enough, you'll find an exception to the common pattern. Over here, Hostoria is multiple, Hostorium is a single one, and it's where one cell extends into the plant cell, and then there's a plasma membrane between the two, but it's absorbing the nutrients out of the plant cell and harming it. This is what a powdery mildew does. It doesn't actually break the cell membrane, but it steals the nutrients from growing on the top surface and then makes more spores and spreads them. So changing the humidity can often help a lot. There was something with baking soda that's a recipe for home gardens is usually found on the internet for powdery mildew. But if powdery mildew goes on, you know, the plant looks pretty bad. You are decreasing the. Now this, do not memorize any of these. I just wanted to show them to you one time. Again, here's the common name. So technically these would actually be in the group called protozoa because they have cellulose, but the water molds, you've probably heard of Phytophthora, which literally means plant destroyer. And then Pythium is commonly found on soil roots around here. Pythium has really round structures, sporangia, and Phytophthora has lemon-shaped ones. And they're very distinctive. So if somebody brings this in and I look under the microscope, I can usually say that, but the people have a lot of work to try to get less water. You know, it's harder to control. And Phytophthora, the species infestans, cause late blight on potatoes because it was unusual that it spread on the wind. Most of these spread in the soil water. Deuteromycetes is a fancy name saying, you know, it's kind of making a judgment to say that they're imperfect. You know, in this day and age, I would say they're different in that we don't know where their genetic recombination comes from. So basically it's like strawberries or taro or anything you're just taking a cutting, it's carbon copy cloning of the parents. There is no genetic, this looks like aspergillus spores. And you can sometimes find aspergillus or penicillium on citrus around here, you know, that gray-green stuff. And all those spores are looking for a little break that they can start colonizing. So they usually can't get through a healthy, intact surface. But if they find a chink in the armor, they're clones and they just keep growing when the growing's good. Now, a lot of times there might be a name here and a name here, or a name here and a name here when they find different forms of the same fungus and give it two names, and it makes it very confusing, and then they change the name. So that part about just know that if I'm telling you it's confusing, you don't have to even worry that. Just, you know, find the most current that you can and use that, or say the old name is this, they've changed it to another one, but in old literature you see this. So it's kind of, there's little discs. Sometimes they have little eyelashes on them. They have little sacs in here that will forcibly eject spores on quiet mornings. So they're called sac fungi. The one like this is, like I said, a discomycete is the actual name for the group. And I thought that was fun because I grew up in the 70s. And then basidiomycetes, basidio is a club. So on the underside of these mushrooms, they have these little club-like structures with 4 little spores that fall off and then blow in the wind. And so, you know, if you don't see these, there's no fungus. No, there's mycelium in the ground. In fact, they had an article saying that an armillaria that grew on tree roots covered acres and acres and could have been, you know, one of this really large organism because it was genetically the same at all these places on that acreage. So actually, if you go get a rotting log and you pull it apart and see the white stuff, that's a fungus in its natural habitat. And it's probably a basidiomycete. And we didn't even hit on slime molds. You know? But here you go. Black leaf streak on bananas, possibly what's over here. And I would say with confidence, Microspirella muscicula. That's what I would say. And I actually don't even know what's the best control for it. We'd have to look it up. But on this next one, we have a different Septoria tritici is the imperfect name. Microspirella graminicola is the perfect name that shows the fruiting structure. So this side here is carbon copy clones that rain splash spread from wheat to wheat. Now over here, and it often happens in the fall, is where there's genetic recombination, like in a flower. And then these ascospores, see it says autumn, land and probably overwinter and have a better chance of survival. But a lot of these well-studied plant diseases will have a cycle where you can see how does it work for leaf spot on wheat. You know, the winter, sometimes winter wheat is sitting there, and you know, what starts it in the springtime, and then what keeps it going, and then how does it overwinter. It gives you, if you break the cycle at any point, you won't have disease. And, you know, late blight on potato that I talked about, this disease cycle goes really fast. You know, some diseases go much slower. You know, virus does not spread from one to the other as fast. So that part is like, how long do you have to work with to make management decisions? So if you look hard, there's going to be lovely disease cycles on the internet.edu things, and then you have more information to work with. But you don't have to memorize any of these unless you work with them like I do. Botrytis cinerea is how I say it. Cinerea is probably another way of it could be said. But they look like little grape-like clusters under the dissecting scope. And they glow just beautifully. You know, you could see little gray dusty powder, but if you have a healthy one right next to it, it's a weak pathogen. And improving air circulation does wonders. So gray mold is common. You might see it on flowers as well, but it's not hard to get the upper hand. This one, black spotted rose, I'm sad to say I have a little rose plant and it lost all its leaves. Sometimes when you see this yellowing, there's a toxin that's produced by the fungus that comes along. And at a certain point, plants say, you know, rather than keep giving up the nutrients, just drop the leaf. And like I said, leaf rust on coffee, 20% of the leaves covered, it drops generally. So my rose lost all of this, but one little branch came back and it's still, and what I did was I changed where I put it, where there was more air circulation. So that part of, I don't have to throw this plant out. It's not a virus. I just have to work to get it in a better environment that's less conducive for the fungus. And, you know, I could take the leaves further away from the plant so they decompose faster before they get on another healthy leaf. Powdery mildew tends to be species specific. So this powdery mildew of papaya is not likely to jump over to poinsettia. So it's kind of nice in some ways, but once it gets going on that plant, and there's a certain, it's high humidity, but usually not quite free water. So powdery mildew is stealing a lot of nutrients. And again, it's closer to gray mold. You can get the upper hand. You do not have to throw out the plant. So we have peppers, we have the poinsettia there, and actually, I'm not quite sure, but usually powdery mildew, you see it more often on the top, but in some cases, it's also on the bottom. It depends on the species. So that part of, and possibly you could see it under a hand lens. And it's powdery, but there's not a lot of necrosis there. So chlorosis is when it loses chlorophyll. Necrosis is when it turns brown and dies. And in this particular one, you see more of a slowdown and less of a death. There are other things where you see plant death, necrosis faster. You know, I use both words when you're talking to somebody. Are they more comfortable with, you know, the cells died or necrosis? Either way works. You know, listen to who you're talking to as to which words you want to use. Brian, I believe, worked on this Phylosticta for his master's degree. And if I understand right, the little spores are all over the place in a greenhouse thing. So if you're growing commercially, if the little spores land and hang out, and then if there's a cloudy day, you know, other things, then all of a sudden they produce symptoms. So isn't it better to prophylactically spray to prevent that initial infection if you have a large crop?
I think they tend to do stuff like that in ornamental production.
And it literally is very reasonable.
Yeah, Phylosticta is a disease.
So that part, you have to be on top of it. If it's like this, you can't save that plant and you might be jeopardizing the others. You know, the plant might live through this, but it won't look pretty and you can't sell it. So Phylosticta, I had a hard time finding it under the microscope, but.
Well, it's always very, on orchids at least, it looks like that pretty much every time.
I think that some of these little pepper spots might have been at the end of Phylosticta, and then some of them were other stuff. So this was a leaf that I just kept it to see what it would do, and this is what it went to. And obviously it's trying to tell me what's going on, and I wasn't getting all of it, but I wanted to show you some of these names because you might see them come into them. I have 5 minutes, and I actually am close to the end. So Fusarium is, there's a lot of species, and then even within a species, there's a forma specialis. And the forma specialis, some can be highly pathogenic and others are just saprophytes. So I can say it's Fusarium, but I can't say if it's killing healthy tissue or just living on dead tissue. But it is common, and it is the bane of trying to grow basil. You know, it gets in the vascular system and it dies. Alternaria is usually spots on a leaf, and it has a little toxin that goes with it, so it makes a yellow halo. So I don't mind Alternaria quite as much as Cercospora. So Cercospora and Colototricum, nice lovely names. Cercospora is around, think of it more like gray mold. It's not going to get the upper hand easily. So if you improve the plant conditions, the plant itself can usually prevent it from causing big problems. But if coffee plants can get Cercospora, and that tells you that you need to modify the nutrition or something else. And sometimes the lower leaves or the shaded leaves or something like that you have less control over. Colototricum, as I said, it's everywhere. Doesn't always get the upper hand, but as in the case of my crown plant, it just keeps coming back, so that plant doesn't thrive. And it's not the plant's fault. And it's, you know, do I want to make it in a more weed-free area or more wind in the area, not planting it next to a wall? So these are names of agents of fungi that cause disease locally. These are common names for trying to describe the symptoms. Then common names are flexible. Is it a leaf spot? Is it a blight that kills more than just the leaves, the stem? A wilt, you know, the plant falls over, it starts in the stem first. A canker is a slow growing spot, usually on something that's woody. You know, a rot is whatever, wherever. Rapid ohia death, Brian was actually in on figuring that out. And it was actually from people that brought in a sample, said, what is this happening? And so that part of Brian had a long list of things that he saw for the first time in the time that he worked here. And, you know, the part of, can we jump on these faster? Anthracnose is a common name for the disease. Colototricum gliosporoides is how I say it. And it is one species. There are more, and they do slightly different things. And these are those spores that I can't quite, they're not distinctive under the microscope. But this little gooey that's kind of peachy colored. And then down here, it certainly got the upper hand of whatever the little.
But it doesn't always come out as peachy, does it? So in the top right picture on mangoes, it's just normally that black spotting.
So that is the first symptom. And sometimes
Oh, but then you'll start to see the peachy happens.
you may, you know, and if you see the peachy, then, you know, that's going to go to the bottom of your compost or the transfer station, or you're going to be spreading it wherever it's at. You know, for potato growers, like, where's your pulp pile? You know, for home gardeners, where's your compost? Something like this should be covered so it can't spread in the wind. It's plumeria rust. Again, it's unsightly, but it's not so bad. Ohia rust. Now, the rust is not rapid ohia death. This kills little seedlings more often. And it came in before rapid ohia death. But again, mountain apple and some other relatives are just devastated by it. So if you see it, there's an explanation, but this is one where we can't shift the balance in favor of the plant very easily. Close resistance is the best bet. Coffee leaf rust, one of the first pictures. And then this was a root one, that this long skinny thing was distinctive cylindrocladium, I believe, or cylindrosporium. There's two of them, and now I forget which is which, but it was a root disease that it kind of sneaks in and slows plants down. That, all of these things, just this is the thug. It doesn't sneak, it just knocks the plant over. So a Phytophthora, a plant killer on taro, and it wasn't around a long time ago. It probably came from other Micronesian islands. So the taro that was originally grown here for many years has very little resistance and usually dies. There are varieties from other places that are more resistant, but it is hard culturally to plant varieties from outside and not be able to distinguish which came from Hawaii for a long time and which came from someplace else. So this is very problematic, but you also want to be very respectful when you talk about it. There's no simple solution. And there's the little lemon shaped. And what comes out of here is these little swimming spores. So leaf blight needs water on the leaves to spread. It's going to be an invasive species. I am now at 1201, so I'm just going to scroll through these. I think you already know this. It's hard to be on an island because we have so many new things, and you are on the front lines. You can help educate people to lessen this. Do not take it outside the states. Do not move it inner island. Like even a ohia tree, it does not leave my yard. Just when you work with people, it's as much of an art as a science. This is a Queensland longhorn borer that is a relatively new pest. If you see these, it's really big. There are some other longhorn borers that have other spots on them that are okay. But the ARS would like to collect living specimens of these. So if you get one, you might want to call ARS and say, do you need any of these right now? Mine actually went there. So I like this, repeat and learn. And garden, the root word for garden and the root word for paradise are the same thing.
Oh.
So your garden is your little piece of paradise. You're talking to people with something that brings them joy. And especially as I was a diagnostician, it's confidential. If I find something on so-and-so's place, it is like HIPAA. And working in the school system, I am not allowed to make anything where people could guess. But I could say, one time I saw this, and just working it back to be that it's a trust and, to be thoughtful and respectful of the people you're working with. We saw this at the beginning, but look, isn't that cute? I just wanted to share. I think it's a baby thrip. Only under a microscope. Entrusted. And this is an adult thrip that I was very happy to find. Whether that was the cause of the problem on the sample I got, I don't know. But the decay and harvest, what's the difference? Decay is when something else does it. Harvest is when we do it. So that part of, in the big scheme of things, nature is not actually caring who harvests what. So it's just we as a society project that it's bad when decay harvests something before we do for the energy. the end.
Thank you.
I didn't leave as much time for questions as they should, but that part.
Yeah.
never-
Well, maybe we'll do some small question portion. Well, I want to open up to online first because I want to make sure Maui, Waka, and Fly have gotten their questions in.
And if you try hard, you can email somebody that could email me with questions, but CTAR website, agents, this is always a work in progress. And even telling that to the members of the public is a good thing.
But no questions online yet. Any questions from you folks? All right. Well, thank you, Roseanne.
Yeah.
And thanks, everybody, for coming out.
I like that.
So we see everybody next week. Oh, you look great.
Everybody get the card for Laura.
All right, folks.
Everybody sign it. Yes.